physiology talks about the life processes and how these processes of organisms functions normally on a daily basis. Well, let me discuss to you what is all about microbial physiology. So from the word itself, microbial, microbes, and physiology, if we combine it together, we will be able to define it as the vital life processes occurring in microorganisms. So in microbial physiology, there are certain metabolic reactions that occurs within the cells of every microbes. So for metabolism to occur, it requires, or every microorganism requires, source of energy. It could be fatty acids, could be amino acids, or those macromolecules that are synthesized for the metabolism to occur in microorganisms. So these requirements are called essential nutrients. So in organisms' energy source, we have terms to be discussed. The terms autotrophs and the heterotrophs, the phototrophs and the chemotrophs. So the main two terms are this: the phototrophs and the chemotrophs. When we say phototrophs, they use light as a source of energy, while in chemotrophs, they utilize chemicals as their source of energy. So that's the difference: light versus chemical. When we say autotroph, these are organisms that uses only carbon dioxide as their source of carbon. So they use the carbon itself. Meanwhile, in heterotrophs, these are organisms that use other organic compounds other than carbon dioxide itself. As heterotrophs are those organisms that uses other carbon-containing compounds which can be utilized as their source of carbon. So when we combine together, when we say photoautotrophs, these are organisms that uses light as their source of energy and uses sole carbon itself, photoautotrophs. When we say photoheterotrophs, they utilize light, but they uses other carbon-containing compounds. So an example for the photoautotrophs are the algae, plants, some photosynthetic bacteria, which includes the cyanobacteria. While in the photoheterotrophs, they also utilize some photosynthetic bacteria. In chemoautotrophs, they use chemical, but in chemoautotrophs, the sole carbon dioxide itself is being utilized for the energy source, while in the chemoheterotrophs, they also use chemicals, but they are finding another way to suffice their needs for carbon. And the example for chemoautotrophs are some bacteria, while in the chemoheterotrophs, it includes protozoa, fungi, animals, and most bacteria. Now next is the metabolism. So when we say metabolism, it is all the different and thousands of chemical reactions that occurs in a cell. So when we say chemical reaction, it is also termed or coined as the metabolic reactions. And did you know that metabolic reactions, as we go to our previous topic in our subject anatomy and physiology, when we metabolic reactions are being enhanced and regulated by metabolic enzymes. So what do we mean by enzymes? So if we will recall our lesson before, when we say enzyme, it is coined as the biological catalyst. So enzyme are those proteins which helps or which could either help certain biochemical reactions to occur or to be accelerated. So these are what we call metabolic enzymes. So before we discussed about the enzymes, let me explain to you first the difference of the division of metabolism. So in metabolism, it is generally subdivided into two. First is the anabolism, and next is the catabolism. So when we say anabolism, it is to build. It is the overall process by which cells synthesizes molecules and structures. So it synthesizes those macromolecules such as the protein molecules, the glucose molecules, in order for a certain cell to build something or to be able to form a newly developed structure and such or likes. Meanwhile, another is that anabolism is also referred to as the biosynthesis. And another one is another thing that is that anabolism is a building process that it requires an input of energy. The second category is the catabolism. So if anabolism is to build, in catabolism it is to break. This anabolism, it has to build. Well, meanwhile, catabolism is to break down. When we say catabolism, it is chemical process in which cells break down from a, for example, this is a large molecule. So if the anabolism is to build, catabolism is to break down such these large molecules into smaller ones. This is the large molecules, and these small molecules is the process of catabolism. And catabolism usually results in the liberation of energy. So if these two are taken together, it makes up the metabolism of the cell. So that's it. Simple as that. Remember, anabolism is to build, catabolism is to break. So let's now go to the metabolic enzymes. So how does metabolic enzymes function in metabolism? So I have here a three-dimensional illustration or diagram or figure about how specific enzymes act upon a substrate. So what is substrate? Substrate are the substances upon which an enzyme acts upon. So how would I explain this? So first, so here's a question. How do enzyme molecules work to bring out a chemical reaction? Take for example, wait, how would I combine this one into something? Take for example, this is the enzyme. This is the enzyme. And these two are the substrate. So the enzyme molecule and these two substrate, for example, this is the two substrate. So this enzyme or this protein molecule will act upon the substrate. So it will combine with the substrate. Oh my gosh, it's okay here. So this enzyme will combine into the substrate in order to bring a reaction which is called the end product. So this is now when they combine together, it is now called the enzyme substrate complex. And then, so in this example, an enzyme molecule is combining. So remember this. So in my example, this enzyme molecule is combining two substrate molecules. Since I'm holding two here, which represents the substrate. So these are the two substrate molecules. So the enzyme functions as the key for the two substrate molecules to combine. And when these substrate molecules are combined, it forms into a single molecule since they lock together or fit together. So once a single substrate molecule is formed from the combination of an enzyme plus substrate, it may be broken apart into two to produce two or more molecules. So after the first part of joining them together, it will now break it apart as here. So it is now it produced or resulted into two molecules. So this is now what we call the end product. See, from a two substrate molecules, we were able to form single molecule, and this single molecule is being break down or separated to form two or more molecules. So that's how enzyme works. So enzymes can form chemical bonds between substrate molecules, or enzyme could either form chemical bonds from a substrate molecules, or it can break certain chemical bonds from a substrate molecules. So that's how enzyme works. So in enzymes, we have this term endoenzymes, exoenzymes. We have the apoenzymes. So what are these? So the enzymes that are produced in the cell and remains at the cell, or it only stays within the cell, it is what we call the endoenzymes. Meanwhile, exoenzymes are those enzymes that is also produced within a cell, but it is released from the cell to catalyze extracellular reaction. So if endo stays, then exo leaves. Remember that. Endo stays, exo leaves. Simple as that. Oh no, I almost forgot. So enzymes are composed of proteins. But not all enzymes composed of proteins. Some are also comprised of a non-protein parts. So we have this so-called simple enzyme. So when we say simple enzymes, it contains protein alone. Simple enzyme, protein alone. While for the conjugated enzymes, they contain some portion of these enzymes contains non-protein, and conjugated enzyme is also referred to as holoenzyme. If all portion of those enzymes are composed of protein, in conjugated enzymes, some part of it does not contain protein or has non-protein portion. Take for example, if this is a simple enzyme and this is the protein, all parts of it or the components inside the enzymes are all proteins. Meanwhile, for the conjugated enzyme, only some part of it or some portion has protein. Meanwhile, take for example, on the other side, it has no protein or it contains no non-protein portion. So that's how they differ. So the protein portion is called the apoenzyme. Meanwhile, the non-protein portion is called the cofactor. So, to catalyze a reaction, take note of this, an apoenzyme must first link up with a cofactor. Just what I have just said a little while, cofactors are those non-protein portions. So, what are the examples of cofactors? It may include mineral ions, such as magnesium, calcium, or iron cation. So these are the example of cofactors. Because when we say apoenzyme, it cannot catalyze their own chemical reaction. So it must be joined first into the cofactors in order for a chemical reaction of these apoenzymes to occur. So that's it for the metabolic enzymes and the